I was kind of advised to first of all discuss what were the reasons why CAR T-cell therapies were introduced, and I think we did the analysis of several trials like the locomotion trial, and we found that patients that had received at least 3 prior lines of therapy, including an IMiD, a PI, and a proteasome inhibitor, and were then progressing, had an extremely poor prognosis. So the response rate to the treatment available at that time was around 30%, and the median progression-free survival was only about 4-point-something months. So it was really bad, and it was clear that we needed new treatment options. And then we were very happy when ide-cel came along and the KarMMa study was done, and we saw that in this patient population, which up to now had only a response rate of about 30%, we suddenly got 80 and more % overall responses. We saw that these patients up to now, being in a median progression-free survival of about 4.5 months, now had a median overall survival of nearly 1 year. And the patients that achieved a complete remission had an overall and a median progression-free survival of nearly 2 years. So this was really an impressive improvement. And it was not at the expense of a high morbidity and mortality. We saw some cytokine release syndrome. About 5% of patients had grade 3 CRS, and some patients, about 3%, had a grade 3 neurotoxicity. So when compared to the lymphoma patients where with CD19 CAR T-cell, we see much more neurotoxicity, especially the treatment with CAR T-cells in myeloma was quite mild. And then the second product came, cilta-cel, in the CARTITUDE-1 trial, and the overall response rate was 97%, CR rate of 86%, and the median progression-free survival nearly 3 years. So again, a fantastic new treatment for our patients. With cilta-cel, we have to be slightly more concerned about some late neurotoxicity, but we also learned, and I think learning is something that is very important in medicine, we learned how to better manage these side effects. So with the late neurotoxicity from in the beginning around 5% went now down to below 1%. So again, an extremely effective treatment with a very acceptable toxicity. Then the next step, and that's important for the regulators, was to kind of compare the new treatment with the standard of care treatment. There were 2 trials, KarMMa-3 and CARTITUDE-4, in both either in 3rd to 5th lines of therapy, or in the other one 2nd to 4th line therapy. The new CAR T-cell products were compared to the standard of care treatment, and in both trials it was clearly shown that when compared to standard of care therapy, the new CAR T-cell therapies induced a much better progression-free survival. So there was a huge advantage again in favor of CAR T-cell therapy. The next question is, where are we moving with CAR T-cell therapy in the future? How are we improving on CAR T-cell therapy? And we learned that earlier obtaining of T-cells for CAR T-cell production makes CAR T-cells fitter, makes, if the tumor has experienced less pretreatment, the tumor is more immunogenic, and also because of less pretreatment, the patient can tolerate this treatment better. These are all arguments why we have now moved CAR T-cell therapy to early lines of therapy. And for example, in CARTITUDE-4, we've seen that now we can achieve 99.4% overall response rate in patients with 1 to 3 lines of prior therapy of their myeloma. And this is a response rate we have never seen with any kind of treatment, even in first-line therapy. So I think, again, showing earlier treatment is important. And we could also show that with earlier treatment, we had less toxicity, less hematotoxicity, less ICANS, less late neurotoxicity. So I think that's the way CAR T-cells are going to be used in the future, earlier lines of therapy. And we now have even trials that are using CAR T-cells in first-line therapy. So one was the KarMMa-2 study, the cohort 2C, where we did a kind of consolidation of patients with a suboptimal response to high-dose chemotherapy. And there was a fantastic upregulation of of the responses by the additional consolidation with CAR T cells. So I guess we are now moving into first-line therapy with CAR T cells. In addition, we now learned that some of the myeloma cells can lose the target antigen, so the strategy now is to use different surface antigens to target the myeloma cells. So we have now CAR T cells targeting BCMA, also targeting GPRC5D. We have dual targeting CAR T cells. So this is one of the strategies also to improve CAR T cell efficacy. The second one is to add substances to improve the performance of CAR T cells in the hostile tumor microenvironment. That's by adding IMiDs, by adding CELMoDs, or using so-called TRACs, which are CAR T cells that are producing cytokines to attract additional immune cells to induce a kind of pro-inflammatory cytokine or milieu in the tumor microenvironment, which also helps to improve CAR T cell function. So I think there's a lot of things that we can still improve, and I think CAR T cells is really one of the big promises in the treatment, not only of multiple myeloma, but definitely for patients with multiple myeloma. That was a kind of a subgroup analysis, and we could show that also patients that are lenalidomide refractory can really benefit from CAR T cell therapy, because lenalidomide refractory patients are an increasing number of patients, and we have limited treatment options, but clearly cilta-cel and other CAR T cell products are a very interesting option for this patient population.